First, sorry, I'm not looking at the right. So epistemology and microgenomics.
Good morning. Thanks to the organizers for giving me the opportunity to share some ideas that goes beyond the technological aspect, impressive technological aspect of this symposium. I would like to, briefly advocate for the fact that microgenomics is not just a technological opportunity. Indeed, microgenomics addresses the issues of cellular diversity, the origins of diversity, the roles of diversity. It's obviously one of the main questions in cellular biology, in biology in general. And but strikingly, two of its main epistemic framework are challenged in the recent times. First framework is that cellular diversity is usually seen as the result of a regulated developmental program, and the second pillar would be that multicellularity is usually described as a cooperation between cells of clonal origins. The first pillar is challenged by features of cell competition within organisms or features of randomness and stochasticity in gene expression that have been described in the recent years. And maybe to a lesser extent, the cooperative based on cells with the same genomes is a bit challenged by different features of variation. Even if we don't speak about microbiomes or chimerism, we could speak of point mutation, copy number variation, mosaicism within organisms. So, to summarize, cells that were long supposed to do the same things, in fact do not, and cells that were long supposed to be the same, in fact are not always so. So understanding diversity and the issue of diversity versus identity is an important question in biology, and I guess that microgenomics can help. Actually, this is a very old debate in philosophy. Leibniz, and a debate that opposed many people, but let's talk about two of them, Leibniz and Kant. Leibniz said that two objects can be labeled identical if they have the same properties, which is reminiscent from Plato's universal types. And Kant said that even if two objects have exactly the same properties, they would not be the same, they would stay at least numerically different. So as biologists, should we care of this question? I guess we should. For example, if we have long assumed that a given genotype had been selected in order to generate a phenotype under a certain set of conditions. But what single-cell studies have extensively shown, and microgenomics does too, is to show that there are many ways to, many roads to achieve functional identity, and maybe that cells are a little bit more Leibnizian than we could have expected before. If we go a little closer to microgenomics, these are two word clouds that I have generated by using reference literatures on genomics and on more recent, of course, microgenomics. You can see that these two clouds are interesting. I would say that the first one, genomics, deals with genes, genome, and DNA, which is basically information, that everything can be put in a computer. And microgenomics seem to deal with a material object, like a cell, obviously, and the way cell uses the so-called information. This is no surprise for you, of course. And if I was to over-interpret a little bit these two word clouds, very raw data, I would say that they depict two different ways of being identical or different. The first one, the genomic one, deals with information, with precision, with regulation, and gives rise to abstract accounts of identity, where microgenomics deal with matter, with constraints, and also with variation and randomness, and course, a different way to be the same, and that could be called a probabilistic similarity. And we can go further and show that microgenomics can address a broad number of questions. For example, it can span from technical issue to very abstract issues, and here clockwise the different disciplines in between, biological and epistemological one. For example, the question of difference versus identity is related to how many cell types do we have, for example, and biological question, differentiation, obviously, signaling networks, what is stereospecificity. It can also impact epistemological issues, like the fact that biological objects are not stable, so what does it mean to be the same when you are not stable, or and the fact that experimental science needs typologies, even though if these objects are dynamic. And we can go also to the more abstract question that I've already addressed. We can translate that into questions. And if we single out one question per field, I would say, microgenomics can help in a way to count, to tell us if there are some hundreds of cell types or if there are as many cell types as there are cells in a way. And if differentiation ever achieved as a stable state, is the fact that biological objects are not stable matter or not in a given biologic context, and to what extent, for example, two objects can be identical. So this question that microgenomics can help you to rephrase or to redefine can also help us to move back toward and to redefine and to reinterpret what we call difference and identity in biology. In order to finish, I would like to end with a sort of very short case study, which is a broad field of stochastic gene expression. I guess I do not need to convince you that a large component of gene expression is stochastic. This has been heavily documented in the last decades in eukaryotes and in prokaryotes. But this is the fact, and there is the interpretations of the fact, and there's still a lot of debate regarding the idea, the role, the functions of stochastic gene expression, whether it is background noise coming from an otherwise adapted fine-tuned system, or is it a biological parameters that can generate a useful spontaneous heterogeneity, like exploratory behaviors or selective dynamics or competitive dynamics within an organism. So of course, microgenomics can help to answer the biological question, but in doing so, I guess it can also help to address more abstract issues, like is biology order generating disorder, or is biology disorder generating order? And this is a very important way to deal with biology, I guess. So to summarize, I would say that obviously microgenomics is a technical opportunity to refine our knowledge at lower levels of biological order, and it will produce fantastic results. No doubt about that, but it's also a promising tool to challenge our representation and to disentangle them from the biological facts. So there are many paths to follow. Thanks very much.
If you measure a certain number of variables in a cell and you measure that in another cell and they look identical, there still may be hundreds of other variables that you can't measure or you are not sensitive enough to measure. You might say that those two cells are identical, but they're not at a different level. And how deep do you have to go before you say that they're identical? This is a problem in tissue analysis because we have experimentalists who look at two pieces of tissue and they measure in the proteomic world with a certain level of sensitivity that's fairly low, only looking at high abundance proteins. And so they see that both pieces of tissue look the same, and they think that that means that there's no change due to, let's say, having the tissue sit out in a hypoxic condition after being removed from the patient. And so that lack of getting down into the depths of the measurement system, into the low abundance analytes, causes us to make inappropriate conclusions about two things being identical when they're really quite different.
I agree.
So how approach you the problem of the order generating disorder and disorder generating order? From my naive perspective, I would favor disorder generating order because it fits, in my opinion, better to evolutionary aspects, because it could offer ways to achieve new states that are of advantage for the progeny later on.
Oh, again, I would say that I would favor, well, of course, I would favor also the second one, but let's not be schematical. I mean, you have some issues where it's interesting and it's useful to start from the first hypothesis, but if you favor the second one, you have to remember that a lot of the knowledge on molecular biology is directly inherited from the first one. I guess we are used to describe the order that is present in the biological system as a produce of a preceding order. And we are used to deal with the disorder that is produced as a background consequence, as some noise, as something that is just the proof that it's not robot, it's biology. But we do not tend in molecular biology to give so many space to disorder as we are used to do in evolutionary biology. And this is why I guess microgenomics deals with every day with variation. I don't know if it's disorder or randomness, but at least it's variation within so-called similar cells or similar tissues. And I guess they will unravel a large part of variability and maybe account it for a disorder, and then maybe this could help to be used in order to describe accurately the way order is produced in a given tissue, not only by the accurate and precise outcome of a program, I would say.
These questions are very, very important, and I am not sure we are able to answer those yet, but we will need to. I'll give you an example. A couple of years back, we essentially took a cell culture and we picked individual cells and we profiled those. And they vary very much in expression profiles, and you can tell from where in the cell culture you picked the cells. And that was related to microenvironment because it's different access to oxygen, you have waste products, you have different neighbors. But that doesn't necessarily, in my mind, make the cells really different. So it's a matter also of defining difference. So my question to you is really, because what this is, I think, is very, very critical, and that's a cell type. How do you define a cell type?
I would say I would define the cell type, to my opinion, the cell type that's a group of cells that are interesting enough to address the function that I'm looking at as a biologist. Like, you know, it's also driven. It's kind of driven by the result I expected to achieve. I guess there's no the problem of the type is a huge problem in biology, in science in general. But I guess there are no types. There are no types. Types are first approximations that are useful for us to study and to try to start to understand a given pathway. But I guess there are no types.
Well, I think we should probably move to the next talk. I could contradict you for now. So, all right, so thank you very much. We'll switch to the next short talk, which is from Dr. Bertu on cell encapsulation for multiplex culture on a chip.